So let's get back to some revision, and so that we will understand where we left off, and also it will help us to remember a few things, right? So in the last class, we were talking about prokaryotes, right? So hold on. So while we were talking about prokaryotic cell, we really discussed some of the specific things about the structure of prokaryotic cell. Hold on. Yeah, sorry. Structure of prokaryotic cell. So now we will try to just have an overlook at what we read in the last class, and then we will continue studying something new today. Okay. So while we were studying prokaryotic cell, we understood that they are primitive cells. They were the first formed functional cells on this planet. So they're not much advanced. Pro means early, carry-on means nucleus. So these organisms are called as primitive nucleus containing organisms, or primitive organisms, what we call as. So while we were studying the structure, prokaryotic cell is no less than an eukaryotic cell. It performs all the functions like that of an eukaryote, but only some characteristic features in the complexity of how the organization of the cell is there. Then it makes a little bit of difference. Okay. So while we studied this, we understood that prokaryotes have cell wall, and the cell wall is made up of peptidoglycan. And below the cell wall, we have is a plasma membrane, and the plasma membrane is semi-permeable. That is that very similar to that of eukaryotes. While we were studying cell wall, or the entire layer, what we call it as, that is cell envelope. The cell envelope is made up of three layers: glycocalyx layer, which is either soft or it is rough, and we can identify the strains of bacteria depending upon the type of glycocalyx that is present on the cell wall. Below that is the cell wall layer, and the cell wall of a bacteria is called as peptidoglycan or murin layer. Based on the composition of the peptidoglycan, you can just differentiate the bacterial cell based on gram positive or gram negative by performing a gram staining technique, correct? And then we have plasma membrane, and we'll study the structure of plasma membrane. All right. So then we understood that the foldings that are present within the plasma membrane of the bacteria, it has a unique function, and we call this inner foldings of the plasma membrane as mesosomes. So these mesosomes, what they do is they perform a similar function to that of the mitochondria. Okay. So and sometimes you have vesicles, tubules, and lamellae, okay, as the foldings. Next one, within the foldings there can be pigment containing membranes, such as in prokaryotes, example is cyanobacteria, right? And one of the most important characteristics of the bacteria that we studied is there is something called as extra chromosomal genetic material. And this extra chromosomal genetic material, we call it as plasmid DNA, which is a double-stranded circular DNA, correct? And I also gave you the details about how the plasmid influences the cell characteristics, correct? Next one, we studied about flagella, the structure of flagella, and the three regions, important regions, that is the filament, hook, and the basal body, and where it is present and how it is organized to function, we study. So villi and fimbria later on. And next, one second. Yeah. Next, we have to study from ribosomes. See, one of the most common features that is between the prokaryotic cell and the eukaryote is the ribosome. Okay. Hold on. I have a message. Sir, could your voice be clearer, please? Okay. Lakshadweep, Jainvi Lakshadweep. Now, can you hear me well? Students, are you able to listen to my voice clearly, or is it the problem for only JNV Lakshadweep? Can you confirm? Clear. Okay. I think Lakshadweep, guys, you have the problem with your network. Hmm. Close other tabs and keep only the Google Meet. Okay. Clear. Okay. For Alaka, even for them, it's not clear. I'm using a microphone, so there is no volume adjustment here. Yeah. Guys, you just check your network, close other tabs probably, so that will help you. If the problem persists, don't worry. will send you the recording of this class if you missed out some things. And I hope it gets better and you can listen to the class. Okay. So I hope I can continue. Okay. Let's go. So now, while we're studying about bacteria, one of the most similar feature that we have in prokaryotic and eukaryotic cell is the presence of a ribosome. But the type of ribosome, what is a ribosome? A ribosome is a macromolecule, and the majority of the ribosome is made up of protein, and the rest of the ribosome is made up of RNAs. So the combination of this protein and RNAs makes this macromolecule a ribosome. So the function of a ribosome is to help the cell to synthesize proteins. Okay, so they do this. So how do they functions? There might be like lakhs of ribosomes within thousands of ribosomes within a bacterial cell. For example, in a prokaryotic cell, there might be around 70,000 ribosomes that are present within the cytoplasm. All right. So now, when we talk about the type of ribosome, okay, when we talk about the type of ribosome, hold on. Yeah, nice to know that it's sorted. Okay. Yes, students. When we talk about the type of ribosomes, even the ribosomes can be differentiated based on their what do you tell? We call it as Wetzberg unit. So Wetzberg unit is based on the density gradient of a macromolecule. So in simple terms, I'll tell that let us say that we have a bacterial cell. I hope you can see the ink. If you cannot see the ink, let me know. Okay. So we have bacterial cell, and then we have a human cell. So now what I do is I take my cell, I extract it. Okay, I dissolve I digest most of my cellular components. Like if there is a cell wall or any cell wall, I'll digest it. And all the biomolecules when we studied that our cells are made up of lipids and it is made up of nucleic acids and so on. Okay, sorry, and so on. So what I will do is I will just choose to not to target any other thing, but I will make sure that ribosomes are remaining. So if I subject this, fluid which contains my macromolecules into a centrifugation, if I can do a centrifugation, let us say that I spin at 10,000 RPM for 15 minutes. Okay. So when I do that, what happens is my ribosomes they separate based on their molecular density. So they separate into two different fractions. One is a higher sedimentation coefficient, and other one is lower sedimentation coefficient. That means if I tell that my ribosome can divide into two separate layers, one layer will be lower, one layer will be higher. Okay, in a tube, after I perform the centrifugation, I'll get two layers, one in the lower bottom, one in the higher bottom. So what does it suggest me? It suggests that some of the ribosomes which are settling at the lower level, that means they have high sedimentation coefficient. Sedimentation means what? Settling down. So that settling down process happens based on the centrifugal force that is applied on these molecules. So we put it in a centrifuge machine. So the centrifugal force influence this molecule or a macromolecule to settle down, okay, according to its specific gravity. So when it settles down, it gives us two layers. So one which has higher sedimentation coefficient is called as larger subunit, and the one which has low, which is at the above, okay, at the upper layers, this is called as smaller subunit. So in bacteria or in bacteria, especially in prokaryotic cell, if you look at it, we have two subunits. One is a larger subunit, other one is a smaller subunit. If you see what is the sedimentation coefficient of the smaller subunit, it will be 30s. What is a larger subunit sedimentation coefficient? That is 50s. Yes, represents the sedimentation coefficient or Schwarzschild constant, where we can differentiate the subunits of not only subunits of ribosomes, any other macromolecule, but since we are trying to differentiate our ribosomes. Our ribosomes have two subunits, 50s and 30s. See, what happens is the function of ribosome depends upon both the subunits. I mean, we require both the subunits to perform a function. So what type of function are we talking about? I told you that the role of the ribosome is to synthesize proteins. Okay. Now, how does it perform this job of synthesizing proteins? You'll study in the next year in molecular basis very detailed. But to give you an idea of what is happening here, just understand that we know that the genetic material is DNA and RNA. Correct, right? So usually in most organisms, the genetic information is stored in the DNA. So whatever the information is stored in the DNA, the necessary information, let's say that the information which is functional or information which is expressing, for example, my eye color, my hair color, or my height, my characteristic feature. So all these components are characteristics which are stored as genetic information in the DNA. Now it has to tell the cell to perform in such a way that it results in whatever the characteristics I see, right? Or do not see. So that is one. The stored information which is in the form of genes are decoded or translated into an RNA. RNA can be a messenger RNA which carries a message from the nucleus to the cytoplasm. When it carries this genetic information message from nucleus to cytoplasm as an RNA, now what happens is these ribosomes, the smaller subunit and the larger subunit ribosomes, what they do is, okay, let me take this new slide, okay, so that I can write. So we have the smaller subunit and the larger subunit. You have the small subunit, you have the larger subunit. So the smaller and the larger subunit along with their RNA together form this functional unit called as polysome. Okay, polysome. So this polysome is what synthesizes the protein. Understood? So the ribosomes in prokaryotic cell, if they ask, it is 70S. You know what is S now? That is called as sedimentation coefficient. So how do we get 70S? Usually the 70S contains two more subunits. One is the larger subunit is 50S and the smaller subunit is 30S. So they combine together to form 70S. Now I know that what you're thinking, why it is not adding. Why are we not adding here? 50 plus 30 is 80. Why are we writing it as 70? The reason is the Schwarzschild constant is not additive. Additive, what is additive and what is non-additive? Additive, you know, like 3 plus 2 is equals to 5. That is additive. Non-additive, why it is not happening here? Because when you have a larger subunit and then you have a smaller subunit, when these subunits come and they join together, they undergo some structural, rearrangement. When they undergo structural rearrangement, now its Schwarzschild units reduced to 10. Understood? When they combine together, their structural integrity changes. The way they are arranged changes. When they are arranged changes, the density changes. So it becomes quite lighter. So that's why we get 70S. Understood? Whereas if we talk about eukaryotes, what's the difference between the ribosome of a prokaryote and an eukaryote? The ribosome of a prokaryote is usually 70S, whereas in eukaryote we have is 80S. But there are some exceptions when it comes to the ribosome. Ribosome are a very important factor to understand the evolution also. You will understand it in the later sections, no problem. So when we talk about eukaryotic 80S ribosome, it is mostly in the cytoplasm. Understand this point. So there are other regions also, and these regions, let me say, for example, if you take plant cells, we have chloroplast. Okay, if you take plant cells, it is chloroplast. And if you take animal cell, even in a plant cell and animal cell, we have mitochondria, right? So what happens is, if you see in chloroplast, the chloroplast in plant cells, which is an eukaryotic cell, the ribosomes is 70S, whereas the mitochondria in eukaryotes is also having 70S ribosome. Some of the animal cells have the mitochondria, which is around 58S or 55S ribosome. Okay, sorry, I will write it again. The mitochondria of an animal cell is 55S. I don't think you can see this. I'll write it here, students. 70S and 55S. You can see the difference, right? So even though there is large amount of cellular structural variations between prokaryotes and eukaryotes, what you can understand is, yeah, the prokaryotic ribosome is also 70S. Eukaryotic chloroplast is also having 70S. Mitochondria is also having 70S and 55S. So there is a similarity in the ribosomes, but you can also see the difference. Is it clear, students? Are you able to understand? Let me check on you. Screen is not clear. Is it true? Students, are you not able to see the words or letters? I don't know how late you're receiving. You're visible. Okay. Network students, please check on your networks. Okay. Did you understand? Can we go for next? Yeah. Let's go. Because your responses will be very late. I will see your responses. If you have any doubts, I will come back to you. Okay. So next one is inclusion bodies. Okay. Next part of the prokaryotic organelles is inclusion bodies. See, what you have to understand about inclusion bodies is they are non-membrane bound vesicle-like structures which are just floating in the cytoplasm of a prokaryotic cell. So whenever you listen to the term called as inclusion bodies, one major aspect that you have to remember is it is non-membranous. Okay, reserve material. I mean, like it's non-membranous bodies that are found in a cytoplasm. Now, what do these inclusion bodies contain? They contain reserve material. Okay, basically, for example, you have phosphate granules, cyanophycin granules, or glycogen granules, okay, food granules, and sometimes also gas vacuoles. And you can find gas vacuoles basically in some of the cyanobacteria like blue-green algae, okay, blue, green, purple, and algae or photosynthetic bacteria. So these are called as inclusion bodies. What you have to remember: inclusion bodies are non-membranous structures which contain either reserve material or they contain gas bodies, okay, which are found in the cytoplasm. Next one. Next, we are going to understand the eukaryotic cell. See, eukaryotic cell is very complex in its nature, in its organization, and how it functions. It is very advanced. Millions of years of evolution has resulted into tremendous amount of engineering. Okay, so this is the best example of how complex a small structure, microscopic structure can be. Okay, this world is seriously. And okay, so when we talk about eukaryotic cells are found in protists. Okay, and it is found in fungi. It is found in plantae, and it is found in animalia. Except for monorans, all the kingdoms have eukaryotic cells. Now, the characteristic feature of the eukaryotic cell is to have membrane bond organelles. Okay, so this double membrane system or the membrane bond organelles enables the cell to compartmentalize its role. What do I mean by compartmentalize its role? It separates its inner environment to the outer environment. Now, if you take an example of mitochondria, or if you take an example of chloroplast or a vacuole, okay, now what you can see is it is surrounded by the membrane that separates its internal environment to the external environment. So this separation enables, uh, you know, the cell to perform its function at a very high efficiency without any wastage and in a very organized manner a cell can function. Okay, and eukaryotic cells are very different in structure and function based on where it is present or what is its function. Now, they also contain cell wall. When you talk about fungi or when you talk about plant cells, you can see that uh, there can be the presence of cell wall in these organisms. Understood? Whereas in animal cells, what you can see is there is absence of cell wall. Another important difference between the plant cell and the animal cell is if you talk about the plant cell, you can see that there is a clear presence of plastids such as chloroplast, chromoplast, and leucoplasts, right? So you might be aware about chloroplast, right? So it's okay. So these are called as plastids, and you cannot find plastids in the animal cell. Okay, 1 second. Yes, JNV Haveri, please tell me. You raised your hand. All right. So leave your message if you have. Okay. I'll continue because there might be some lag. Okay. Chalo. So, sorry. Okay. So I was talking about the difference within the eukaryotic cells. And as I told, I was also talking about the plastids that are present in the plant cell, that is chloroplast, chromoplast, and leucoplast. It is not found in the animal cells, right? We don't need that because we are heterotrophs mostly. Another significant difference, what we can find with respect to our plant cell and animal cell is, see, in plant cells, we don't have centrioles, whereas in animal cells, we see the presence of centrioles. This is another difference. Okay. Now, eukaryotic cells are complex. They have cell wall, they have complex cytoskeletal structures and other structures which help this cell to move also. They are motile cells. So the motile cells are also very highly organized. If you look at a typical eukaryotic cell, what you can see is these are some of the structures which we call it as cell organelles that are found in eukaryotic cells. Okay, so now, for example, if I take an eukaryotic cell, a typical plant cell, what can I find? So I can find cell membrane, cell wall, endomembrane system. Within that, it includes endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, and then we have mitochondria, plastids, ribosomes, cytoskeleton, cilia and flagella, centrosome and centrioles, nucleus, and microbodies. Okay, the list is big, but we can study the list in a very simplistic manner. Okay, and similarly, the same things are found in eukaryotes, but what you can see unique in eukaryotes is the presence of centrosome. Okay, apart from that, all the structures are same, but the absence of cell wall is clearly seen. All right, students. So now, when we talk about the cell, okay, especially the eukaryotic cell. Now, I told you that eukaryon defines, it translates to new nucleus or eukaryon. Why it is so important that we classify the cell based on the nuclear organization? First thing is because if you look at the cell functions, most of the cell functions are controlled by the nucleus, which is in the form of genetic material. And in eukaryotic cell, what you can see very unique thing is the entire genetic information is organized in the form of chromosomes. Okay, very important thing, that is chromosomes. So this organized chromosomes which are present within the nucleus are separated from the cytoplasm by a double membrane, okay, structure called as nuclear membrane. Now, this double membrane structure, which is a nuclear membrane, is very, very significant, very highly significant to really differentiate the cytoplasmic environment to the nuclear environment. Now, here, with a clear distinction, what happens is whatever the genetic information that is sent in the form of mRNA, it goes through this nuclear membrane, and then here, with the help of endoplasmic reticulum and ribosomes, the manufacture proteins and the cell functions will take the place as it is. Okay, so the organization of the genetic material in the form of chromosomes, presence of a nuclear membrane is highly significant characteristics of our eukaryotic cell. Okay, so as usual, first we will start studying a typical structure of a eukaryotic cell by starting with cell membrane or plasma membrane. Very interesting structure. Okay, 1 second. So we can't see clearly. What's the problem? Give me a second. Let me check on you. Yes, students, this is blur and this is super sharp. I think it should be visible. Yeah. In the screen. Okay. No, like visibility in what sense? And it will be visible for students. Okay. All right. Okay. Is it only for the Haveri or confirm with students? Okay. Later in the chat box. What you can't see? You can't see the board clearly or the letters clearly. or the entire thing is blur, so let me know. Okay, it can be the upload also, it will take a little time. All right, so now we will start studying with uh, the cell membrane. Hold on, letters are blur. Okay, hold on. So give me a second. About now, hold on. I think it's visible now. Okay, sorry students. Okay, I don't know whose fault it is, which side. Okay, so it can only be resolved like this. Is it everybody feels that the letters are very blur? Because on the screen I can see it's very clear in the camera, but I don't know how it's visible for you people. So if everybody are feeling that you are not able to see the screen even now, please confirm. If not, we will see. I'll just try to fix something visible. Okay, thank you. I hope it's visible. Okay, so now it's very very different situation for me. Hold on. Yeah, so clear. Okay, thank you. Let's go. So we were talking about starting with cell membrane. Very interesting structure, students. Okay. So before we start studying the cell membrane, we have to take a little bit step back to understand the biochemical nature of the cell. So when we talk about the biochemical nature of the cells, one of the most important things that we studied in biomolecules is lipids, right? Lipids. Hold on. Okay, we talked about lipids, and in lipids, one of the important types of lipids is phospholipids, right? Hold on. Sorry, it's not writing here. Okay, phospholipids. So in phospholipids, what is the nature of phospholipids, students? Lipids are hydrophobic molecules, correct? You agree with me? All of them are hydrophobic. But when we talk about phospholipids, they are unique. They are not water fearing. They act like an amphibian. That means they love and hate water at the same time. That's why we call phospholipids as amphipathic molecules. Why phospholipid is an amphipathic molecule is its structure. We know that a typical lipid has a glycogen, and to this glycogen, you have two of the carbon atoms, okay, are attached by an unsaturated fatty acid chain, and one of the carbon atom it gives rise to the phosphate molecule, phospholipid, to a three carbon atom. Right? Can you hear me? Sorry, students, there was a technical error. The Wi-Fi went down suddenly, so I didn't know, I didn't realize where we stopped or what's the last thing you heard. Can you just help me out with that? So, no. So, anyways, I think we're starting with plasma membrane. So again, I'll go with plasma membrane. So what else about? Okay, I hope I have an folder. Clear, clear. Phospholipids. Perfect. Okay. So coming to the phospholipids, nature of the phospholipids, as I was saying, they are amphipathic molecules and they have two important regions. One is a hydrophilic and hydrophobic. And I was talking about the hydrophobic nature. Hydrophobic nature is this two carbon hydrocarbon tail that we have here. Okay. So hold on. Two hydrocarbon tail. And you have your phosphate head which interacts with water freely. Okay. So that is why what is the most important characteristics of your plasma membrane or your phospholipids. See, if I generally ask you a question, then the question is, students, what is the composition of a plasma membrane? With my experience, I'll tell you the most general answer I will get is it is made up of lipids. Correct. So this is what the generalized answer I get. Yes, you are right that the plasma membrane is composed of lipids, but to what percentage of lipids are present in a plasma membrane? If you look at the composition of the plasma membrane, what we can find is the lipid concentration along with the concentration of proteins varies with respect to the function of the cell. For example, if I take a red blood cell, 55% of the red blood cell is made up of your lipids, whereas 40% is made up of proteins. Okay, so that is a composition of the plasma membrane. You understand my point? So the composition of the plasma membrane, that is the lipid to the protein ratio, changes from one cell to another cell. For example, we have taken red blood cell, that is human erythrocyte or red blood cell, and we check the composition of the cell membrane, we can find that it is made up of 55% of lipids and 40% of proteins. Now, let us talk about the arrangement of lipids. Now, if you look at the plasma membrane and ask you to define the structure of the plasma membrane, what you will do is the plasma membrane is a lipid bilayer structure. So you use the term called as lipid bilayer structure. So that means a plasma membrane is made up of essentially two important structure, correct, right? Two important layers. So you have an outer layer and you have a inner layer. So let's see how it is. So you can see that let us assume that you have one lipid layer and you have another lipid layer. So they are just overlapping on each other like this. Okay. Now what you can see is the lipid layer has two ends. One is a phosphate head and it has a unsaturated fatty acid chain or a tail. Okay. So this phosphate heads, we call it as hydrophilic heads. Okay. So this is called as hydrophilic heads. Hydrophilic heads, and then you have the chains of your unsaturated fatty acids which project towards inside. Okay, so all these chains are projecting towards inside. So this is hydrophobic core. Understood? So here this is phosphate, here it is phosphate. Now you can see that here it is a hydrophobic core. Now what happens is the interaction between these chains, okay, the chemical nature of these chains, what it does is they will be attracted to each other so that they will never slide away from each other. They will always have this force of attraction that is binding them together. Understood? Now, one of the other important characteristics of the plasma membrane, apart from having the arrangement of your hydrophobic and hydrophilic ends separately, is between this tail, that between these layers, there are some proteins and cholesterol molecules that will help this plasma membrane to slide over each other. So that is what gives a plasma membrane a quasi nature. Quasi nature means the ability of this plasma membrane to slide over. So this characteristics the plasma membrane gets. Understood. Now we have the nature of the plasma membrane, where outside layer where it is having the phosphate end, okay, is hydrophilic, and then what we have towards the inner projection, which we call it as hydrophobic core, we have, correct, right? Now I told you that between these two layers there might be cholesterol and proteins which help the plasma membrane to slide upon each other. Now, why such an arrangement is very, very important. When scientists, when they performed an activity to understand the nature of the phospholipids, they dropped it in a water. So when they dropped phospholipids in the water, the phospholipids naturally they gain this micelle formation where they start arranging in such a way that they create a new environment within this bubble. For example, the all this black thing that you see on the screen, assume that it is water, and then assume that I am dropping some phospholipids. When I drop some phospholipids, what I see is the phospholipids are arranging themselves in such a way that they are trying to protect all this hydrophobic or water non-interacting, or the molecules which do not freely interact with water, towards the inside. All this inside core is hydrophobic and the outside heads are hydrophilic. So this kind of an arrangement, a very basic chemical nature of these phospholipids enabled a lot of possibilities for the living organism. How? With the creation of this structure which can really isolate the external environment to the internal environment. That is the gateway for separating the external environment to the cellular environment. So one of the very first structures to be very well defined is plasma membrane in a cell. Okay, so the plasma membrane is very, very active, and it is a semi-permeable membrane. Semi-permeability means it allows only what it needs and it does not allow what it does not require. So the semi-permeability of the plasma membrane is very highly regarded to its nature and its composition. We understood by its nature that the hydrophilic and hydrophobic interactions are quite Students, I'm extremely sorry. I hope you can hear me. Oh, hold on. Yeah, guys, you can hear me, right? Were you students there for the rest of the class, or with the class stop because of this network? Please let me know, okay? All right. So we will meet tomorrow, students. Okay, I will just try to get the information till where you listen properly or where you could not, and then I will continue from there. Okay, I'll repeat it. All right. Thank you very much for your patience and for understanding the technical difficulties. See you tomorrow. Thank you.